Hello and welcome everyone. Thank you so much for attending the afternoon session. We are really excited to have Dr. Kapil Bharti speaking to you all about during this session. So welcome to the Advancing Cell Therapy session. Today's session will provide an overview of stem cells and how they can use to save or restore vision. My name is Chad Jackson. I am the director of the Preclinical Translational Research Program at the Foundation Fighting Blindness. I would like to review some logistical details for this session. Currently, all participant lines are in listen mode only. Today's session is being recorded, live streamed, and available in closed captioning. To activate the closed captioning, please select closed captioning at the bottom of the interface. If you are using a screen reader, please be aware the controls are at the bottom of the Zoom interface. This control bar may collapse when not in use. If you prefer to prevent the controls from auto-hiding, go to the settings within the Zoom platform, select accessibility, and then select always show menu controls. To ask questions during this session, you can use the Q&A feature on the Zoom control bar and then type in your question. All sessions during the conference will be recorded for your viewing later. Before we begin, I'd like to encourage you to be social with us. Please share your experience in using the hashtag Vision2020 on Facebook and Twitter. Now it's time for our main event and for our featured presentation. The speaker for this session is Dr. Kapil Bharti. Dr. Bharti is a senior investigator in the Ocular and Stem Cell Translational Research Section at the National Eye Institute in Bethesda, Maryland. Dr. Bharti holds his PhD from the Johann Wolfgang Goethe University at Frankfurt in Germany. I'm sure I probably mispronounced that, so Dr. Bharti, you can correct me once it's your time. His work there involved basic biology in the areas of heat stress, cellular chaperones, and epigenetics. From Germany, Dr. Bharti came to the National Institute of Neurological Disorders and Stroke, where he published numerous papers in the areas of transcription factor regulation, pigment cell biology, and developmental biology of the eye. It's perhaps this combination of diverse backgrounds that led him to develop an interest in the emerging field of stem cell biology, particularly of the retinal pigment epithelium. He moved into the role of staff scientist. Dr. Bharti has authored numerous publications and has won several awards, including being a finalist in the prestigious NIH Earl Stedman Symposium. With that being said, the virtual floor is yours, Dr. Bharti.
Thank you, Chad. Thanks for this kind introduction, and I want to thank Foundation for Fighting Blindness for organizing this session and for inviting me to present some of our work and also kind of give an overview of the stem cell therapies. So let me try and share my screen, and we are live. So as Chad mentioned, I'll give you a brief overview of the field of stem cell and stem cell-based therapies and how we and others are using them to treat eye diseases. As many of you are aware, vision loss really affects many different aspects of patients' life. As you can see on this slide from left to the right, it affects night vision in many patients, causes night blindness. It causes in many patients kind of a tunnel vision where they lose peripheral vision and only can see in a small portion in the middle. And in many patients, they actually lose the opposites. They lose a central vision and high acuity vision, ability to distinguish colors. And I listed example of some diseases here like Bardet-Biedl syndrome, choroideremia, retinitis pigmentosa, Best disease, macular degeneration, some of the diseases that we are studying in my lab, where we see that many of these symptoms happen at the same in patients' life throughout their life, one at a time, or multiple symptoms happening throughout their life. But the sad part is that worldwide, there are millions of people affected by various different kinds of eye diseases, and it looks like there were 300 different types of genetically manifested eye diseases. And unfortunately, for many of those diseases, there's really no treatment. So it's really debilitating for many patients. So for all of these diseases, or for most of these diseases, the part of the eye that they affect is the back of the eye. If you view here, this is kind of at the bottom of the screen, you see the schematic of a human eye. The light passes to the front to the lens, the cornea, the lens, and then hits this back of the eye called the retina. And the central part of this retina, where we have our highest acuity vision, is called the macula. This retina is composed of a homeostatic unit between several neurons. A key neuron of this is the photoreceptor, which is a light sensitive cell, and it's present next to a monolayer of pigmented cell called the retinal pigment epithelium cells, which is sandwiched between the photoreceptors and the blood supply. And these three cell types form really the homeostatic unit in the back of the eye. And the reason I say that is anything goes wrong with one of the three cell types, and this is what happens in most of these 350 diseases I talked about, or I briefly mentioned, is that the other cells start to suffer. And of course, as eventually when the photoreceptors would die, patients would go blind. Most interesting for my lab is this cell type called the retinal pigment epithelium, which is sandwiched between the photoreceptors and the blood supply. And it's really there for a strategic location, for a strategic reason, is because this cell type throughout its life performs several functions to support both the photoreceptors and the blood supply. So in cases where retinal pigment epithelium cells die off, the photoreceptors would suffer, as you can see on this slide, and also the choroid blood supply would suffer. But really, in many different diseases, there are examples where photoreceptors would die first, which leads to eventually RP death and eventually the choroid death. So, or if the choroid would start to die, it would also lead to degeneration of the RP and the photoreceptors. So really different disease stages and different diseases manifest the loss of one or the other cell type in the eye. Just to give you an example of what kind of approaches have been tested to kind of treat or to slow down disease progression in some of these cases. For instance, if you look at this slide, there's this area called, in case of macular degeneration, this area is called the transition zone, where from completely dead cells to cells, photoreceptors that are perhaps better, there are photoreceptors that are destined to die. These photoreceptors are not functioning properly. As you can see, their length is shortening, and that's because they don't have the RPE support, which is missing here. And what clinicians have tried is transplant RP cells and try and rescue these photoreceptors. And different approaches have been tested. And this next schematic actually shows you an example of how we can use different approaches to try and treat different diseases. So this is, if you think from the left side to the right side, a normal looking retina, different neurons in orange, green, blue, and then the retinal pigment epithelium. As the tissue starts to disrupt, we can try to treat this by gene augmentation and gene editing. And the perfect example we know is of LCA that has been recently treated with gene augmentation for RP65. But as the cell types start to lose in the retina, and the example that I just showed you of a loss of RPE, we need to come up with a replacement treatment, because once the cells are gone, no gene therapy or no small molecule or no antibody can treat that. And I'll give you an example today of RP patch that we have developed to treat patients with macular degeneration. Now consider a completely disarrayed tissue where we have lost more than one cell type. Actually, we lost more than one tissue here. In that case, we may need to replace more than one tissue that have been gone. So RP with photoreceptors or RP with the choroid blood supply. And I'll give you two examples of our ongoing work, very preliminary data of trying to replace RP and choroid and RP and photoreceptors in the cases when we have this disarray in the late stage diseases. So this is going back to that example. Let me start with this idea. And this really is a slide that we have made for how we are going to treat patients with macular degeneration, AMD patients, where we hope to transplant an RP patch, which is shown here in purple, on its own Brooks membrane. That's the membrane that the cells sit on, in this area where the photoreceptors are slowly dying. And interestingly, this approach has been clinically tested in many different patients. And what surgeons have tried is they would cut out, I don't know if you can see my hands, if they would cut out a piece of RPE from the periphery of same patient's eye and translocate that into the center. And in small number of patients, very small number of patients, about 5% patients, where this RP patch would engraft, they would see that the photoreceptors above that the transplanted RPE would be stable. But you can imagine this is a complicated surgical procedure, often leads to a lot of hemorrhage in the area where the RP is cut out. So the engraftment rate is very low. But it really formed a guiding principle for the work that we're doing, that if we can make such an RP patch from AMD patients or for any patient, we may be able to treat the disease or have the disease progression slow down. Then the question comes, how do you make such a patch? You know, in the surgical procedure, they would cut out from the same eye. But if we have to make it in a dish, the only way we can make any patient's tissue is from stem cells. So what are stem cells? A stem cell is a cell type that has two key properties that you have to know. One is that it can replicate itself. That means it can make more copies of itself and almost indefinitely. And second key property is that it can make other cell types of the body. It can make neurons of the brain, it can make blood cells, it can make any epithelial cells, it can make other stem cells. So it can really be differentiated in a dish to any other cell type of the body. There are three key stem cell types that are known. The embryonic stem cells, these are the cells that are present inside the pre-implantation blood cells actually that make us, that make the whole body. These cells give rise to us. And the second cell type would be what is called induced pluripotent stem cells. These are the cells that can be derived from any adult person's cells, but they behave in very similar to all of their properties to embryonic stem cells in all practical purposes. And then the third is tissue-specific stem cells that you may have heard of, cells that make our blood. They renew our blood every couple of months. Stem cells in the cord blood, and cells in our fat tissues and other areas of the body. These stem cells, the third type, are more specialized. They can only make a handful of cells. For instance, the blood stem cells can only make blood cell type, and they do that throughout our lives. For the purpose of this talk, I'll focus on iPS cells, the induced pluripotent stem cells. Pluripotent term means these cells, again, can make any cell type of the body. How do we make iPS cells in a dish? We can start, as I said, with any somatic cell of a person. So we can start with the small punch from the skin, a little bit fibroblast isolated from the skin, or we can isolate blood cells by a very small blood draw and reprogram those cells in a dish using a handful of factors called the reprogramming factors that were discovered about 10 years ago and led to a Nobel Prize for two people. And these cells, these reprogramming factors, in about 3 weeks of time, would convert these skin cells or blood cells into iPS cells. And then, as I mentioned, these iPS cells then can be differentiated into your favorite cell type, blood cells, gut cells, or cardiac muscle cell, or in our case, any of the eye cell type that we want to make. And that brings me back to what I'll show you today, is that the approach for treating patients, late stage patients with degenerative eye diseases, would be that we start with the patient cells, reprogram them with these reprogramming factors into iPS cells, and make either retinal pigment epithelium, the photoreceptors, or the choroid with its all intact vasculature, as you can see here, and then bring back this tissue to the patients, and hopefully that will work as a treatment for late stage diseases. Now, keep in mind that if we make patients on cells, if you start with patients on cells, we may have to remove the diseased gene. And that can be done with the gene editing techniques. In vitro, there's a number of techniques available, and many different labs worldwide are trying those techniques. So it's really, at present time, this is a very feasible pipeline. I'll show you that we have done this pipeline once for macular degeneration patients by making RP cells. But making photoreceptors, choroid, and gene editing, all of that is currently feasible. So just before I go into the details of the data, I want to give you a sense for what it takes to do such a clinical trial, what it takes to bring such a cell therapy to patients. The whole pipeline is described here, from doing basic research to really getting approval from the FDA so that the technology is commercially available for patients. It takes somewhere around 10 to 15 years, and it depends how long the basic research have been. It can be even longer. The different parts of the pipeline is from basic research to, we go into what is called the translation research, where we do what is called IND enabling studies. That means we develop an investigational new drug application to get approval from the FDA so that we can start a phase 1 trial. And I'll show you a lot of data today, how we got an IND approved for a phase 1 trial for macular degeneration patients. And then it's really going through phase 1, which is a safety and feasibility study, to phase 2 and 3, which are your pivotal efficacy studies, to getting the product approved by the FDA. And all of this is during this time, we are monitoring the safety, first in animals, then initially in patients, and then we're looking for whether in a larger population, the drug, or in this case, the cell therapy, has any efficacy. At this stage, sorry, go ahead.
I'm sorry, can I just interrupt one second? I think that there's a question here that just help you, I think, clarify your point on this. I think some are interested in knowing sort of, is the retina being regrown in the body, in the person's body, in the eye, or is it being regrown in addition, being placed in? And also, could you sort of weave that in with the severity of a disease such as RP? When would you foresee this therapy being available, in sort of at what disease stage would somebody have to take advantage of this technology?
Okay, thank you. So to answer the first question, no, the retina is not being grown in patient's body. This is very important. It's a very key point, because we're not transplanting these stem cells in patients' eyes. These stem cells, iPS cells or ES cells, also have the potential to become tumorigenic if they're purely transplanted. So very important point is that in a dish, we take these iPS cells and make the eye tissue in a dish, and then we transplant pure eye tissue without any iPS cells. And we go at length to demonstrate that there's no iPS cells or stem cells left in our final tissue. And it's purely eye tissue. And I'll show you that we also check whether the tissue is actually functioning properly in a dish before we would transplant it in patients. And the second question was what disease stage? And I think, let me take you back actually to a slide where I show you. I think these kind of treatments would only be applicable to late stage diseases. So here's an example, even back, let's say. Here's an example of late stage macular degeneration, where, as you can see, the retinal pigment epithelium is gone, the retina is gone, and the choroid is gone. So when tissues are going to be lost, that's what we will bring replacement tissue. It's considered a replacement therapy. If you are at an earlier stage disease where the tissues are still there, but they're not functioning properly, I think the approach one would want to take is either gene augmentation, enhancing the function of genes, or repairing the genes that are defective so that the tissues are not lost, so the tissues start functioning. But as the tissues or cell types get lost, the only option we have left there is to start replacing them one at a time or in combinations. Any other questions so far? All right, so I just went over briefly the timeline for putting together such a clinical trial or getting approval for such a cell therapy. At this point, I also want to take a moment to distinguish between what would be an FDA-approved therapy versus what is an FDA unapproved therapy. And I think this is a very important distinguish, because there's a lot of what is called snake oil out there, and I want to warn the patients to be very careful of not going after the snake oil and be actually, that may cause more harm than benefit to them. And here's an example. These places often are called stem cell clinics, but in many cases, they are selling unapproved and unproven stem cell to patients as potential treatments. And here's an example where 3 patients actually lost their vision after intravitreal injection of so-called autologous stem cells for AMD patients. These are macular degeneration patients. So patients should be very careful of asking the right questions when somebody is offering them a stem cell treatment. Has this been FDA approved? Have they gone through this kind of a pipeline? Even if it's a phase 1 trial, have they gone through this IND approval process? If it's a later phases, what phase it is, how many centers, who is leading PI, who's the leading center on these kind of studies. These are very important things patients should be careful about. And some places, just I can suggest where there's more information available. Of course, the FDA, our Food and Drug Administration, has a website where they provide very clear information on what kind of therapies are, stem cell therapies are FDA approved and what are not. NIH has a web page where they also provide similar information, and International Society for Stem Cell Research, ISSCR, has also a web page where they provide very detailed information on what are approved versus unapproved stem cell therapies. And just a couple of pointers I want to give. If somebody wants to pay you, wants you to pay out of pocket for a stem cell treatment, it is likely not approved. Because if it is a trial, then the site or the sponsor will pay for all your treatment. And if it's a commercially approved stem cell treatment, then your health insurance should be able to reimburse you one way or the other. There are mechanisms that this can be done either through Medicare and Medicaid or through insurance. And then currently keep in mind that there is no FDA approved commercially available stem cell based treatment for the eye, for the back of the eye, for the retina, RPE, or the choroid. So these are a couple of pointers patients should keep in mind as they're looking for options for themselves. So going back to what I started a few minutes ago, how do you actually bring an RPE tissue? How do you make it in a dish, and how do you bring it to patients? Again, just as a small introduction of what are the contents of an IND application. So this is a first step to get approval from the FDA to start a safety phase 1 study for any treatment, and in this case, stem cell treatment. We have to demonstrate to the FDA that we can make that eye tissue in a dish. We can make it reproducibly, we can make it safely, we don't have any xenocomponents, we don't have any contaminants in it. This tissue is sterile for all the times we make it, and we can consistently make it. And then we have to demonstrate to the FDA that the tissue, when transplanted in relevant animal models, it will not lead to any tumor or toxicity. And when transplanted in a disease model, animal model again, that it will have an efficacy effect in that animal model. And all the tools, all the reagents that we use, we have to demonstrate that they are biocompatible. So there's no reaction of patient's body to any of those tools. And then of course, we have to have our clinical protocol to get approval from our institute's IRB board, institutional review board, and have the patient's consent to this trial. So I'll walk you through how we make this tissue and also some of the data, how we prove in animals that the tissue is safe and it works. So again, it's a very busy slide. Don't worry about the details. This is a very quick overview of how a very simple tissue, which is a monolayer of cells, the retinal pigment epithelium, RPE, is made from stem cells. We add these different growth factors. Again, the names are not important. Just remember that these are growth factors that are used in the body, that are used as the tissue is made, as used by our body to make the RPE cells. We have chemically obtained these growth factors from companies, some of them made by ourselves, that help these stem cells to become mature RPE cells in a dish. So we follow step by step and we monitor the cells as they become RPE cells, that they are going through the exact stages that they would normally in a human body that to become the right RPE cells. And at the bottom is two examples here. On the left side, you see a dish full of pigmented cells. As many of you may know, RPE cells, the name stands for retinal pigment epithelium. These cells are pigmented, similar to the pigment in our skin cells. So we can see them in a dish, they have darker color. And then we show by various mechanisms we use that these cells are pure. Here's an example. All these little dots are showing that these cells are pure. If they're above in this quadrant, that means they are all pure cells. More than 98, 99% purity we can easily obtain. And that's critical because we don't want to transplant any stem cells in anyone. And then of course, and another example here would be that the protocol is reproducible. We have to show that we can make it from many patients, different type of patients, healthy volunteers, different disease conditions. And all of this was done to provide to the FDA that we can reproducibly and consistently make this tissue. How do we actually make this tissue? So once the cells are made, how do we want, because the tissue has a monolayer architecture, but the cells are in a dish. So we take the cells from a dish and we put them on this scaffold. In this case, this is a scaffold made of a biodegradable material called PLGA. In full form would be polylactic glycolic acid. Again, name is for different purposes, different material can be used and different material is being used. But the point is that use of a material allows us to make a tissue in a dish, and the material degrades within a few weeks. So it's not staying in patient's body. And what it does is these nanofibers, as you can see here in this electron micrograph of this tissue, they allow the cells to stay on top, form a monolayer, and make their own membrane, that Brooks membrane that I was talking about, on which they sit. And then the tissue degrades and the scaffold degrades, and we are able to transplant the tissue. So this is how the cells look like once they grow on top of the tissue. Each hexagon is a cell. There's different colors, and excuse me for people who can't distinguish between green and red. This is how the stainings are done. They are often done using green and red colors, but the yellow is showing you the borders of the cell. Each hexagon is one cell. And you can see the more hexagonal they are, we know closer they are to human RPE cells. This is a very detailed process of how we make cells in the clean room. For transplanting in patients, we can't just make them in our lab. We actually transfer the process into a clean room where they have HEPA filters and other means to ensure that no dirt particles go in, no microbes can go in, so that the tissue doesn't get contaminated. So it's a very special process, and every step of the process is documented, and literally on a daily basis, we document dozens and dozens of paperwork to make sure that we know that every step is done correctly. This process, again, details are not important, takes 6 months for every patient to make their tissue, starting from day 0 when we collect blood, when we make the stem cell bank, when we start converting that stem cell bank into the eye tissue in a dish. It takes about 6 months. But the important point is that we have quality controls measured at every step to ensure that the process is going in the right direction. The stem cell banks, we characterize the genomic stability, we characterize if they are the right kind of stem cells. We make sure that before we start using them for differentiation into eye tissue, we make sure that these banks are right. And then when we are making the eye tissue, like I said earlier, we know every developmental stage of that eye tissue. We check by relevant markers, by transcription factor and other markers, that these eye tissue is being made correctly. And finally, when we have our RPE tissue, again, we confirm its sterility, its purity, its identity, and its function before we'll transplant in patients. This is an example of how our eye tissue, the RPE tissue, looks like once it's made from stem cells. And I hope you remember the schematic where I showed you that these RPE cells have this little hair on their head. And you can see here, this is a very high resolution image taken by electron microscope, where we can see the hair on each cell. So this is one cell, another cell. So these are different cells. And you can see all the cells have the similar looking hair, suggesting they all differentiated similarly into this RPE tissue, into this eye tissue, and the membrane that they made is shown here, and the scaffold is shown here in the bottom. So I won't go into details of all of these assays, but we have made this now, at least in case of AMD, which is our first trial from 3 different patients, and we made 3 different banks per patient, and we differentiated them all into eye tissue and characterized them by various different assays to really understand how consistently we can make it, how much is the variability from person to person when we make this eye tissue and when we functionally characterize them, and what are the actual functional characteristics of the tissue we can use to show every time we make it that we are making the right tissue. And this is really, really important. I cannot stress enough that we make sure that stem cells are gone, we make sure that the tissue is characterized functionally and structurally before it's transplanted. And this is an example. This work is published, so I won't go into too much of the details, what kind of assays were done. Now, once I've shown you that how this tissue is made, how it's characterized, again, as I mentioned that for the FDA approval, we also need to demonstrate in animals that the tissue is safe and has efficacy. So to demonstrate that it's safe, we need, remember we're testing human tissue. So if we transplant human tissue into an animal's body would reject it as a foreign object, right? It's immune rejection. So we have to use special animals called immunocompromised. That means their bodies cannot reject human tissue. So we transplant this in a rat eye, and we monitor this tissue now for almost 9 months. And we've seen that for 9 months straight, the tissue doesn't cause any toxicity, doesn't cause any tumor or doesn't leave the site from where we transplanted. And here is an example. In red here on top here, you see human RP cells transplanted. And actually they are nicely adjacent to the rat RP cells, and to the point that they integrate themselves into the same monolayer as they were rat RP cells. So they work so safely, they integrate so safely into the rat eye. And this preliminary work, based on this preliminary work, we did a very large study at a CRO, at a contract research organization based in Madison, Covens, where we tested almost 450 rats that were transplanted with either a small tissue, 0.5 millimeter. Our human dose of tissue would be 2 millimeter by 4 millimeter, but the rat dose would be about 0.5 millimeter circle, or the actual human dose of cells, which are 70,000 cells on this 2 by 4 millimeter patch. We transplanted either the 0.5 millimeter circle or the 70,000 cells, and monitored these 400 rats for over 9 months and collected samples at 2 weeks, 13, 26, and 9 months, and to determine that the cells and the transplants were safe and they didn't cause any tumor or toxicity. So then to test that this transplant can actually function in an animal model which has a similar disease as we see in patients, we tested it in pigs. We used pigs because, as I showed you in the previous slide, this transplant is relatively big. It's about 8 square millimeter. There's no smaller eye we could use. We could not use a rat eye to transplant 8 square millimeter piece because the eye is way too small. So we needed an eye that was big enough as a human eye and similar in anatomy to human eye, where we could test this piece. At the same time, we needed to optimize the surgical procedure, how we will deliver it to the back of the eye, right? So for all of the reasons, we use pigs as our model system. But in pigs, unfortunately, there's no genetic model that leads to a similar disease as macular degeneration. So we generated an injury model that causes a similar damage seen in the back of the eye as is seen in macular degeneration patients. And here we're using a micropulse laser that damages the pig RPE cells by heat. And as the RPE cells die, the photoreceptors would die, similar to what happens in patients. And the idea would be then we would transplant the human RPE patch here and have it save the pig photoreceptors, so that we see that the transplant is actually functioning. So as an example, how it looks like, this is on the left side, you see a pig eye fundus photograph. It's infrared image. Normally the infrared light would not pass through the eye, but when we laser damage the pig RPE, it passes through that area. It's a 7 by 4 millimeter area of pig eye that we laser damage, and you can see this, what we call the window defect, where the infrared light passes through. So we know that this area of the RP has been damaged. And then we can do histology on that eye to show that actually the RP is indeed damaged. And here you can see the monolayer of RP is missing in this area. And also the photoreceptors are dying. This is, if you look at this situation, this is very similar to any late stage retinitis pigmentosa or macular degeneration, but it's an acute condition we have created by laser injury. And the idea now would be transplant the human RP patch here and have it protect the pig photoreceptors. So to do that, we had to develop a tool that we can use to deliver the patch. Here's the tip of the tool here in the bottom corner you see, and you can see this RP patch in here. This is actually an empty patch, but you can see the patch inside the tip. This is how the patches will be delivered. And then this cannula, the tip of the cannula is connected through a tubing through a foot paddle, so the surgeon can control it and maneuver the delivery of the patch. Everything is filled with hydraulics so that the maneuvering of the patch is much easier. So all of this had to be optimized, had to be designed to fit the patch, to fit the curvature of the back of the eye, and have it biocompatible and safe for delivery of this tissue to the patients. Here's a brief video. So disclaimer, I'm going to show you a brief video of the pig eye surgery, but as you can see, the eye has been prepped here. A hole in the eye wall is being made bigger so that in the next shot, you will see the tool inside the eye. Here's the tool, here's the RP patch. You can see pigmented cells in the patch. the retina has been lifted and the cut in the retina has been made bigger, and the surgeon is aligning the patch, the tip to deliver the patch under the retina. And then the retina is flattened and the patch stays right there. And then again, for us, it's very important to monitor that the patch is being delivered to the right place and stays at the right place once it's transplanted. But the beauty of working with the eye here, of course, is that we can monitor this by non-invasive techniques. This is a process on the left two side images. You will see a technique called OCT, optical coherence tomography, where we are measuring reflectance from the back of the eye. So we shine light and we measure what changes at the back of the eye. So in the left side, you can see this patch is a bandit-like structure and has a cut in the retina. If you look at it from the side view, and I've marked here with a yellow line, here's a patch here under the retina, and the retina looks fairly healthy on top of the patch. We run it through people who do this kind of work routinely, and they tell us that the retina looks really healthy on top of the patch. But we can also objectively measure that by using electroretinogram of the pig eye. Basically, what electroretinogram is when we shine light onto the eye and we measure electrical response from the retina, because retina is a neuronal tissue that's electrically conductive. But we use another sophisticated technique called multifocal electroretinogram. Basically, instead of shining one light pulse, we can shine many small light pulses to the back of the eye. And depending on where the light will hit the retina, we'll only get response from that part of the retina. For instance, in this right-hand side in the graph that you see, the bottom hand graph shows healthy retina. This is a signal we get from somewhere away from where we did the laser injury. We get a nice and big response. And then in the middle is the area, this would be the laser part here, where we laser injured the retina but did not deliver the RP patch. We get no response. And then green would be the area where we laser injured the retina but deliver the human RP patch. We get a response that is in between the two, healthy and the injured, suggesting that the human RP patch is saving the pig photoreceptors from dying. Again, since we're doing this in animals, we can also prove by histology that there is rescue of the photoreceptors. Here's an example of images from two eyes, from two areas from the same eye, a laser area with no transplant and laser area with the transplant. And you can see the transplant in the bottom here marked with the yellow line. Notice the thickness of this blue line, layer marked by this arrowhead. It's almost double in this area as compared to in this area. This is a layer of photoreceptors. It's almost double the thickness in this area above the transplant, suggesting that our transplant was able to save the pig photoreceptors from dying. So this is where this work was submitted to the FDA late last year and was approved for a phase 1 to a safety and feasibility study to test autologous iPS-induced pluripotent stem cell derived RP patch for a late stage geographical atrophy stage of AMD. We have 2 cohorts for these patients. First 5 will have visual acuity between 2100 and 2500. And next 7 patients will have visual acuity between 2080 and 2500. Now, again, this trial will run for at least 2 to 3 years. Based on the outcome of this trial, we'll launch a 2B and hopefully eventually a phase 3 study. Now, I want to take you back to this, that, and I think somebody asked a question that, what stage are these tissues transplanted? We are only transplanting the RPE tissue, but as you can see in this case, the damage is sometimes that the photoreceptors are missing, and sometimes the choroid is missing. The work that we have done on this RP patch and brought it to patients has formed the basis for us to also build upon more complex tissues and bring them to patients. So I'm going to show you some of the ongoing work now that we have started in the lab with the hope to take that to a phase 1 trial in the next few years. So now I mentioned that this would be a case where there's really a late stage disease, more than one cell type is missing. And I'm going to show you 2 examples, one of transplanting RPE and choroid, and another of transplanting photoreceptors and the RPE. So first, how do we make the choroid? Again, same example, like I showed you how we make the RPE. We start with stem cells and differentiate them into a cell type called the endothelium that makes the blood vessels and the capillaries. Similar process, developmentally guided. We use various growth factors at various time points, make sure cells have gone through their various developmental stages and then become mature cells. We prove them that by various assays, by looking at them through microscope and looking at their purity, that we can make these cells over and over again, make these cells purely pure, and also make them consistently from multiple different patients. Once we have these cells ready, what we do is we mix them with 3 different cell types. So we mix endothelial cells with pericytes and fibroblasts. These 2 cell types form the support architecture around the capillaries, around the vessels, and we mix them with hydrogel to form a bioink. Why do we make a bioink? Because we want to bioprint them to make little capillaries from this tissue. If you don't know what the bioprinter is, works very similar to a 3D printer that many of you have seen. It essentially has 2 needles that move in a very precise XYZ orientation that we can control. And at the bottom, I'm going to show you a little clip how this works. There's a needle going in a zigzag orientation where it's printing our bioink. And we do that because it allows us to make capillaries in this very defined orientation. So the way this works is, if you remember the scaffold that I showed originally that we used to make RP, well, we take the same scaffold, flip it upside down, we bioprint our bioink on one side of the scaffold, flip it upside down, seed RP cells on top, and let the scaffold degrade over time, and the 2 cell types make their own extracellular matrix, which is a Brooks membrane that is naturally found in the eye. This process takes about 5 to 6 weeks. RP cells mature on top, the vessels mature at the bottom, and the tissue starts to look something like this. Here's the only capillary tissue. As you can see, the capillaries are marked here. These yellow structures are capillaries. And each lumen that you see, this tube-like structure, is a little capillary like we have everywhere in our body that carry blood all throughout the body. So these capillaries run from one end of the tissue to the other end. Some are bigger, others are smaller. They range in diameter from 10 to about 50 micron, very consistent to the diameter that we see of capillaries in our body. So this is how the whole tissue looks like. Here's the RP cells on the top and the capillaries on the bottom. And if you go up, so I'm going to go through one more time here. RP cells on top and capillaries at the bottom. This is a 3-dimensional view of the entire RPE choroid tissue that we have generated. And now we're starting to transplant that and tested it in animals. Here's the first example of testing it, because we're making tiny capillaries. We don't know if they're functional. The only way to test their functionality is to transplant them and see if they would carry blood. So here's a capillary, human capillary transplanted in a rat eye, under the rat skin, excuse me. And you can see this red color marks what the human capillaries are. And you can see here's an example of a small capillary that is carrying rat blood. So these capillaries are becoming functional once they're transplanted. They're connecting with the rat blood circulation and carrying the blood from one side to the other side. That's exactly what we want them to do once they're transplanted behind the eye. Here's a very first attempt that we have done to transplant this tissue in an eye. So what you see on the left side is a schematic of our tissue, retinal pigment epithelium on the top, the membrane in between, the capillaries at the bottom, and we show by staining them, by looking at them by their markers in microscope, that retinal pigment epithelium cells are indeed on the top. There's a membrane area, and then the capillaries are at the bottom. And then when we transplant them, in this case, we actually transplanted them behind the eye, and we're looking at through ICGA. It's a technique where we look at, we inject a dye and we look for different blood vessels called angiography, combined with the technique that I showed you before, OCT, where we look at reflectance for the back of the eye. And these little circles here, these dark shadows, suggest to us, we are now still confirming this, suggest to us that the human capillaries are indeed connecting in the back of the rat eye as well, and they're functioning there as well. The last example I want to show you is of tissue that now contains RPE and the photoreceptors. This is the work that we're doing in collaboration with a friend and colleague, Dave Gam from University of Medicine, and his company, Opsis Therapeutics. They have developed a different kind of scaffold where they are actually growing photoreceptors inside. So if you see on the top right corner here, the scaffold looks like an ice cube tray. So each cube has receptors that you can see here that live in it. And there's tiny pores at the bottom of each ice cube is from where the photoreceptors grow out. And you can see on the left here, the schematic, they have this long legs called the photoreceptor outer segments, which is where the business end of photoreceptors is, where the visual pigment sits and how they see. These outer segments would grow out of these pores and then talk to the RP cells that are lying underneath that. So we have obtained this scaffold and the cells from them, and we started to co-transplant this scaffold with our RP scaffold that I showed you before. So here at the bottom is our RP scaffold, if you remember from the first half of my talk, and on the top is a photoreceptor scaffold characterized again for various different markers. And again, the same pig model, and this is the beauty of that work I showed you, that we can leverage it now for developing future more complex therapies, is that the same transplantation tool, same animal model that can be used to test other cell therapies. Again, in this case is a back of the pig eye, same OCT technique that I showed you before. We laser damage this entire area, and you can see this laser leads to degeneration of the retina. These bright spots is where retinal cells are clumping up. And this is an area where we have found where the RPE and the photoreceptor transplants are co-residing on top of each other, the photoreceptor transplant on top. And you can see that here with the bright line. See, the retina looks much healthier here as compared to in this part and in these areas. We have now started to do more sophisticated analysis to actually look for whether the photoreceptors are alive in this transplanted area by a live imaging technique, looking at single photoreceptor at a time. It's a technique is called adaptive optics. So here's on top is the same image that I showed you in the previous slide, much more bigger here, the healthy area, the laser damage area, and then the transplanted area with the RP transplant at the bottom and the photoreceptor transplant at the top. What you see here at the bottom is images taken using this adaptive optics technique, which is looking at single photoreceptor, single cone photoreceptor at the time. Cone photoreceptors are the key photoreceptors that are responsible for our high acuity vision and for color vision. If you see in the healthy area, you see many of these dots. Each red dot is one cone photoreceptors. Our computer software can count them, can segment them, and tell us that there is a given number for each healthy area. Now, if you compare that to the laser area, you see much fewer photoreceptors because our laser damaged them, killed the photoreceptors, and you can see our computer program tells us much fewer dots and much fewer photoreceptors in this area. Whereas if we go to the transplanted part here, and this on the left side, we seem to have restored the balance, restored the number of cone photoreceptors, more similar to the healthy part. And this is within few weeks, within transplantation, within about 4 weeks, we can restore the number of cone photoreceptors in this area. Now we can tell by this technique whether the technique is looking at pig photoreceptors or the human photoreceptors, but whichever it is, we're restoring the number of photoreceptors, and that is key to bringing vision back to these pigs and hopefully eventually to patients. So I want to leave you with this preliminary data and a really hopeful thought that we are at a point that we think we can assemble the entire back of the eye using vasculature at the bottom, the membrane between the RPE, the retina, and finally the retinal blood supply. And hopefully within the next few years, we'll start to bring these treatments to patients. With that, I want to thank the entire team. As you can imagine, this kind of work is not possible by of course, by myself alone or by even a small team. This requires a very large team of people. And here's most all of them, some of them who are not in this picture. Actually, I should tell you that this, for all their efforts for developing this advanced cell therapy, they got an award from NIH director, Dr. Collins. And this is all of us receiving that award. Some of them who are not in the picture are mentioned here or up here. So with that, thank you very much for your attention, and I'll take any questions you have.
All right, thank you so much, Dr. Bharti. Now we'll spend the remaining time taking questions from the audience. We've already compiled quite a few questions already. As a reminder, you can ask a question by accessing the Q&A feature on the Zoom control bar, and then typing your question there. So I think out the gate, the number one question that's been coming across in your presentation is, for example, if someone has already gotten a genetic test, however, it's come back inconclusive for the genetic mutation, would they still be able to receive this type of treatment if it's 100% successful?
Once it's successful, potentially yes. At this stage, it's going to be very hard because, as you know, at the early stage, we have to be very mindful of the safety of such procedures, and FDA mandates that we only transplant it in patients where we know exactly what disease they have, what genetic condition they have. So this is, at this stage, it's going to be not possible. But I think if this treatment becomes very successful and becomes a routine practice, then I think we should be able to get permission from the FDA to transplant irrespective of the genetic condition, known or unknown.
All right, and just to build on that theme, is this treatment going to work for sort of all cases of retinal damage? And if the retinal damage is genetic based, would it also have to be accompanied with genetic therapy?
So if there's retinal damage, either photoreceptors or RP, the treatment would work because it's replacing your damaged cells. If it's a monogenic condition, that means there's a genetic underlying basis for the disease, yes, we will need to correct the mutation if you're starting with patients on cells. But as I said in the introduction, that technique, that process has already been established, and it is easy to correct genetic mutation in situ, that means in a dish. We don't have to correct it in the patient, and then transplant the corrected cells. There's another approach that we are now working on with this company, where we are taking what is called allogeneic cells. That means one healthy cell type from a person and make RPE cells, make retina cells, make eye tissue from that. And that tissue will be available to actually all the patients. The only caveat there is that in that case, since it's somebody else's cells, the patients will have to stay on immunosuppression for a while. And again, that work is ongoing. We're trying to establish how long will that immunosuppression will be. Is it 1 year, 2 years, or 5 years? Immunosuppression, as we know, are strong drugs and they have some side effects. So to avoid that, we started using autologous tissue, but I think at the same time, in parallel, we want to be testing the allogeneic tissue to avoid the need to do genetic correction.
All right, thank you. The next question is, can you sort of intimately describe the stem cell patch and how it integrates into the existing retina? And also, when you talk about the treatment, how similar is it to the normal physiology of the human retina?
That's a great point. Let me actually go back to, if I can find a good slide to show you. So this would show you, let's see if I can. So this is a human RP patch made from stem cells. And as you can see, it's just fitting comfortably under the retina. It fully integrates by this technique. We can compare by OCT, it is there. By this technique, multifocal ERG, we show that it is working as well. If the transplant was not integrated, was not functioning, then we will not see recovery in the retina. The fact that we're seeing the retina is recovering, retina is not dying, is suggesting to us that the transplant is working. Again, I didn't go into details of all the tests that we did, but we've gone through many tests to demonstrate that the transplant functions like the native. So here's an example of these are all the different tests that we went through. And they are, again, it's all published work. I can provide if FFP wants to upload on the website for patients to review. Many different ways we have proven that the transplant functions almost exactly like the eye tissue.
All right, great, sorry. One question we have here is, can you again speak to the level of retinal damage and how effective will this treatment be? So if someone has complete loss of photoreceptor cells, maybe other cell types, is there any limit to where you see this treatment can be actually effective?
So complete loss of photoreceptors, I think this is where we think that this approach would work, where we'll have to transplant the photoreceptors themselves as the scaffold with or without the RPE patch. If there's complete loss of photoreceptors and all the other neurons, I think this is going to be the most challenging approach. But rest assured that there are labs that are working on a 3D retina RPE graft. And I think that approach, it's a few years away, but that approach should work for such patients.
Okay, thank you. Another question is, who else is working in this field, and when do you, if you had to guess, when would you actually see something being available in the clinic as an FDA-approved treatment? How many years out? Who is probably going to bring the technology to bear?
Yeah, good point. So, if I go back to my timeline of how long it takes, so this is our timeline. Our treatment, our trial is at this stage, so phase 1. So we are somewhere between 4 to 8 years from being commercial approval for the RP patch. There are a few other companies that are trying similar approach with a similar RP patch made from either embryonic stem cells or slightly different approaches. So they are also at a similar pace, a similar stage, and they should be 5 to 8 years away from getting commercial approval. Now, the next step of it is the more complex treatments that I talked about, including photoreceptors. That is more at the IND enabling preclinical stage. So that's, I would say, at least 3 to 4 years behind. So again, in 10 to 12 years, we should start to see even those treatments, if successful, obviously, at the phase 1 and 2 getting approved. But the phase 1 of those, where some patients at least can enroll, and phase 2, where at least a few patients can enroll, is about 4 to 5 years away. Many labs around the country, worldwide, David Gam, I mentioned, a close friend and colleague, his company, Opsis Therapeutics, they're doing similar work in Madison. There's a couple of companies in California, where Regenerative Patch Technology is one of the company. Astellas Pharmaceutical has a similar RP patch. So there are many companies, many different labs are doing this work around the world and in the United States as well.
All right, thank you. So could you also give your perspective on, if consider a patient that would have more than one thing to address? For example, if a patient had AMD and may be eligible for this type of treatment, but also had an issue of dealing with glaucoma as well. Would you think that only certain patients would be available for this type of therapy, or would it still be, in your vision, available for all?
I think once we reach phase 3 or approval stage, it should be available for a broader set of patients. At this stage, because we are still assessing the safety profile, we would not want to risk the patients that have more than one conditions. I think because then it would be more complicated for them. They will also continue to need the treatment for glaucoma, and we would not want to kind of mix the two treatments at this point. But once it's approved, it should be available for a broader set of patients.
All right. And I think we might have time to squeeze one more question in. And just a general thought from you, what do you think will dominate in the clinic, genetic therapies or stem cell replacement in the future?
I think both have their own space. Let me go back to one of the slides I was using to explain that. So genetic therapies have their space at an earlier disease stage. If a disease requires that the cells are not functioning properly because of the genetic mutation, and we can do that, we can improve that by gene editing or gene augmentation. So that's a stage when the genetic therapies have space. But if the cell type is gone, for instance, in macular degeneration, the example I showed you, RP cells are gone, or late stage RP, retinitis pigmentosa, where the photoreceptors are gone, or choroideremia, where our retina, RPE, choroid, all of them are gone, would be this arrayed stage. That's where gene therapy has no room. I think that's where we need the replacement therapy. So the difference would be gene augmentation, gene editing, genetic therapies at a slightly earlier stage of disease, whereas cell replacement and tissue replacement at later disease stages.
All right, Dr. Bharti, thank you so much for your presentation. We are currently at time. I'd also like to thank the audience for joining. I think we had over 300 people join this session, so that's wonderful. I'm sure everyone learned a lot. Please join us tomorrow at 11 a.m. for our Promise of Genetic Therapy session. Thank you all again, and have a great rest of the evening.
Thank you. Goodbye.
Bye.